are together, it's difficult for us to open, but using the lock that is usually on the zip, you can easily unwind it. So think of DNA replication like a zipper. When the two zip ends are together, they're held by hydrogen bonds. But you can't replicate DNA if those DNA strands remain like that. You need to unwind them. And when we say unwind, it means breaking the hydrogen bonds. Okay? And then, of course, when you break the hydrogen bonds, you're going to have a region that is going to be open. And that's what is referred to here as replication fork. We're going to look at them in detail, but for now, we are saying that replication has got 3 major steps. The first one is initiation. And the key feature in initiation is unzipping, breaking the hydrogen bonds. The second is elongation. In elongation, you think of this step as the one where we add new nucleotides, meaning that at this point, you must understand the structure of a nucleotide and which end of the nucleotide you're going to use to make another strand. Is it the 3' end? Is it the 5' end? Or in this case, what do we find on the 5' end? It's a phosphate group, right? What do we find on the 3' end of a nucleotide? It's hydroxyl group on the pentose sugar. So that understanding must be very clear here. And then finally, termination. Like any other process, there must be an end. And DNA replication is not unique to that. At the end of the day, when all the information that is a genetic material is duplicated, it must come to an end, to terminate. So we're trying to say DNA replication, the process itself is just 3 steps. You initiate, you elongate, and you terminate. So let's go through each one of them step by step. And I'll start by asking the question, where does DNA replication start? Remember, when we're looking at the features of DNA, we looked at the origin of replication. I could have forgotten, but I think we did. DNA replication has to start from somewhere. Yes, we know it has to initiate, but where does it initiate? And the region where it initiates, what makes it a good region for it to be an initiation site? So we're going to start by saying that initiation begins at the region that is referred to as an origin of replication. Imagine you have a strand of DNA and those strands are being held by hydrogen bonds. Replication doesn't just start from anywhere. It starts at a specific site, and in bacteria, we call it an ORI-C. I think somewhere in the notes. In bacteria or prokaryotes, the region where DNA replication starts or where initiation of DNA replication starts is referred to as an origin of replication or an ORI-C. Okay, just C. What you see in this diagram is that region initially was held by hydrogen bonds, and then those hydrogen bonds are broken or melted. And what you see is kind of a bubble. On either end of the DNA, you still have your double strands together, held by hydrogen bonds. What this means is that when DNA is opened or unwound, or when the hydrogen bonds are broken at the origin of replication, it is not the whole stretch at one time. For example, if this was the initial strand of DNA and this is all the genetic material in that cell, only a specific region, a small region, or a short segment is going to be open. And there's a good reason for that. We can discuss that as we go on. But for now, we're trying to say replication starts at the origin of replication. When replication starts at the origin of replication, keep in mind, it is not the entire region that is open. No, it is only a short segment of that DNA molecule. Now, what are the features of this origin of replication? This, we've already looked at it and we even know why. The origin of replication is rich in AT bases. Remember, you have to break the hydrogen bonds, and the cell actually uses energy, ATP, to do that. And remember, the system is fine-tuned. It is made in such a way that it must conserve energy. So it makes sense that the origin of replication has got a rich TC region. What do you mean by rich T and A? So rich T and A, you're trying to say that region has got more bases that are either adenine or thymine. And we know why. When you have A and T, the number of hydrogen bonds is 2. And the cell can use less ATP, less energy to break those hydrogen bonds as compared to if this region was G and C. Because where there's G and C, you've got 3 hydrogen bonds. So it makes sense that this origin of replication has got a rich A-T combination. And in bacteria, this is how it looks. What you see here is the origin of replication. I want you to keep in mind that in bacteria, the chromosome is circular, or the genetic material in a bacteria cell is circular. What this means is that there should be a circle around this, straight line that I've put there. At this point, my interest is just the origin of replication, but keep in mind, this stretch here is part of a loop, because in prokaryotes, their DNA is circular. Ours is linear. So even though I've depicted it as a linear molecule, it is not linear. Around it must be part of a loop. So let's go back. What features does the origin of replication have that make it suitable for it to be a starting point? We are saying that it has got a rich AT combination, and we know what that means. In bacteria, the OREC has got 2 regions. It has a region that is referred to as 9-mer. The mer just means the number of nucleotides that are there. It has got a 9-mer sequence, and these are in repeats of 4. Like, for example, where there's this first blue, if you can see my red dot there are 9 nucleotides there. And those 9 nucleotides, again here, there are 9 nucleotides, 9 nucleotides, 9 nucleotides. So it has got a region in the origin of replication that is referred to as a 9-mer. The 9 there represents the count of nucleotides in that region. In addition to that, this region also has a 13-mer. The number 13 just represents the count of nucleotides. For example, if I pick the first yellow box, the number of nucleotides in that region is 9. It's 13 in this case. So the 13-mer are only 3 repeats, meaning you've got 13 nucleotides here, 13 there, 13 there. This is one region of the origin of replication. And then you have another region of the origin of replication. This one has got 4 repeats. So it's got a combination of nucleotides there. There's a stretch that there's a kind of like an intersection, like a section which is separating the two. Another one there repeats with 9 nucleotides, and another one there. Collectively, this is referred to as the 9-mer. And this one here, there are 3, is collectively referred to as the 13-mer. Let me say this again. So what are we saying here? What you're seeing here, shown like a straight band or line, is representing a section of the origin of replication, where in prokaryotes, bacteria, this region has got 2 segments. One segment is referred to as the 9-mer. What does 9 mean there? Or just the mer, M-E-R. 9-mer just means the number of nucleotides in that blue section. So for example, if this is a blue section, if you can see the dot, there must be a combination of 9 nucleotides. Again there must be a combination of 9 nucleotides. Collectively, these make up a region referred to as a 9-mer. And we're going to see in the next slide why this region is important and why the other one is important as well. But collectively, these are components of the origin of replication in bacteria, not in eukaryotes, no. This other region here is referred to as a 13-mer. You can see it says 13BP. BP means base pairs. Okay? The tandem array of 3 13BP sequences. There are 3 of them. You can think of them as yellow boxes. And each one of those yellow boxes has got 13 base pairs. Each one of these is referred to as a base pair. Okay? Because remember, DNA is double-stranded. But I don't want to confuse you. What I'm just trying to say here is that the origin of replication in bacteria has got these segments, 2 segments. One is the 13-mer, one is the 9-mer. The 9-mers are 4, the 4 blue whatever boxes, the 13-mers are 3. Now, what happens at these positions? So during DNA replication, or should I say, I don't want to skip, I don't want to go too far. So the 9-mer in your notes are indicated. These are also referred to, this is referred to as the DNA box. The 9-mer sequence, the blue boxes, these are referred to as DNA boxes. And I'll explain in the next slide what DNA A is. It's DNA A. So this is a DNA box. And this one here is a rich AT base pair. So meaning that both of them, they could serve different functions, and indeed they do. So now, let's move on to find out what do these regions do, the 9-mer and the 13-mer. So remember, the 9-mer is a DNA box, also referred to as a DNA box, for the simple reason that it has got the site where DNA A can bind. I hope I'm not confusing you. I'm feeling like I'm not saying this properly. What I'm trying to say here is that the 9-mer region in the origin of replication consists of sites where DNA A can bind. I'll explain what DNA A can bind, but first it's important for us to understand that that region, it has got segments that are referred to as DNA box. That DNA box is just a name given to the 9-mer. So each one of those boxes is like a DNA box. Now, what does the DNA box in the 9-mer do? It allows for the binding of DNA A, which is just a protein that is found in bacteria and is referred to as the initiator protein. The DNA box in the 9-mer of the origin of replication, it acts as a site where DNA A can bind. DNA A is a bacteria initiator protein that begins DNA replication. In short, the cell knows that DNA replication must start at the origin of replication, but who is going to identify that origin? DNA A comes in as a bacteria protein that identifies this region in the origin of replication. That is why the first contact in initiation is recognition of the origin of replication. Are we recognizing the 9-mer or the 13-mer? No, it starts at the 9-mer. The 9-mer is a site that is recognized first, marking that this is the origin of replication. The protein in the bacteria that recognizes the origin of replication, that is the 9-mer, containing those DNA boxes, is DNA A. Now, once DNA A, you can see these initiator proteins there. They are there to recognize the ORIC. But remember, the ORIC has got a 9-mer and a 13-mer. So which one of these is recognized first? It is the 9-mer. Why? Because the 9-mer has got a DNA box. Like trying to say, it has got a binding site for this protein that we're referring to as DNA A. Now, once this DNA A binds to the 9-mer, it causes a conformational change, like a distortion, okay, in that region. And this action, that is to say, the binding of DNA A to the 9-mer, causes local unwinding. A small region is going to be unwound. What this means is that the region where DNA binds in the 9-mer causes a conformational change, it's like you've added, if you've got a thread, and then suddenly you wrap it around a stone. In that region, it's no longer linear, or the structure is going to change. So the binding of DNA A to the 9-mer causes a conformational change, like a stress, and this stress allows the DNA to unwind upstream where the 13-mer is. Remember, the 13-mer is a region that is quite rich in AT bases. The 9-mer region is just there for recognition of the oric, while the 13-mer is a region where DNA must unwind. The 9-mer is just a region on the oric, or just at the oric, that is responsible for recognition of this site, to say DNA must start here. And the protein that recognizes that region is referred to as DNA-A in bacteria. The binding of DNA-A to the 9-mer causes a conformational strain on the DNA, allowing the region upstream, and by upstream I mean kind of in front of the 9-mer, which is the 13-mer in this case, to unwind. Now, at this point, it is important for us to understand that DNA-A is not the enzyme for unwinding DNA. No. DNA-A just comes in one to recognize the site of replication at the 9-mer, and its binding causes it's kind of like a distortion. And because the region up front in the 13-mer is weak, it kind of opens up, a short segment. Now, it is also important for us before we move to realize that replication, we need the templates to be separate. The DNA templates must be independent of each other. They should be bound by hydrogen bonds. So then, to ensure that the region that the 9-mer has unwound in the 13-mer remains single-stranded DNA molecules, the enzyme helicase comes in to continue unwinding the DNA. To stabilize the DNA, the DNA also recruits single-stranded binding proteins to ensure that as the DNA helicase is unwinding, those strands remain separate. They do not, anneal, because remember they're complementary base pairing. The moment you unwind them, they can still go back. But because you want to replicate the DNA, you have to keep them independent. To keep the unwound DNA strands separate, this process of replication recruits single-stranded binding proteins. Let me go to the next slide. Maybe this will be helpful. So now, at this point, before helicase can come in, the DNA must have a region that is single-stranded. Helicase does not bind to double-stranded DNA. It only binds to single-stranded DNA. How does a single-stranded DNA come about? It comes about because we have DNA-A first recognizing the 9-mer. Once the DNA-A binds to the 9-mer, and specifically where in the DNA box, it causes a conformational change upstream in the region where we find the 13-mer, which is rich in AT. This local unwinding, and because I'm saying local because it's such a short segment that is unwound, allows the enzyme helicase to come and bind to one of the DNA strands. So what you see here is DNA helicase. Okay? And in bacteria, it is referred to as DNA-B. Okay? So in bacteria, the enzyme helicase, which is the enzyme that breaks the hydrogen bonds between the strands of DNA, is also referred to as DNA-B. DNA helicase, allow me to just refer to as DNA helicase, but you also know it's also called DNA-B, cannot bind to double-stranded DNA. It only binds to a single strand of DNA. And its function is to break the hydrogen bonds. And keep in mind, this process requires ATP. And that is why, because breaking hydrogen bonds is an energy-dependent reaction, the opening of DNA has been made in such a way that it is not too energy demanding. Okay. Now, I want us to look at this diagram. I want us first of all to pay attention to the way the strands are designated, or should I say just numbered. The blue strand is designated 5'. The green strand is designated 3'. Now, The DNA helicase is bound or attached to the 5' strand and not the 3' strand. Now, this is not a mistake. DNA helicase binds to the 5' strand. When you look at replication, you're going to see that replication, is bidirectional, and polymerase can only attach, can only add nucleotides in a specific direction. But in this slide for me, the take-home message here is that DNA helicase 1 cannot bind to a double-stranded DNA. No. So it only comes in once DNA A has caused a distortion and the 13-mer is partially unwound. When it binds to DNA, which of the two strands is it going to attach to? Is it the 5' or the 3'? It binds to the 5' strand. Okay? And at this point, I want you to also picture this. Remember, this is a bacterial DNA is a circle, meaning that while we have this on this section, we should have the same on the other region, because remember, when it unwinds, we're going to get the whole region unwound. I just went back. If you look at this stretch here, so meaning that there's going to be helicase going this direction and helicase going that direction, like opening up the DNA. So meaning that what you see here is just one section of that replication origin. And at some point, you're going to see that we need you to be able to picture both ends of the replication origin. So now, this is just maybe trying to emphasize what I was just trying to say. We have these proteins here. This is DNA. When it binds to the 9' this region is strained. And because it is strained, it causes a region up front in the 13' to kind of unwind partially. The partial unwinding in the 13' caused by DNA allows helicase to bind. Helicase of the two strands binds on the 5' strand. So now let's look at this. Let's say, for example, origin was here. Okay, so we've got this helicase, and we know the function of helicase is to break the hydrogen bonds using ATP. So it's going to be proceeding in this direction. We also have another helicase here. This one is going to proceed in this direction. And this is what you see here. It's opened up. So you've got one going this way and one going that way. But look at the strands. If I pick this one, if you can follow me, this is a 5' end. Helicase is here. If I look at this strand, this is a 3' end. So then you can see that on this end of the helicase, this is a 5' end. Why? Because we can split this section. That is to say, replication is going this way on this strand, and this strand should also be independently designated as going this way. What I'm trying to say here is that you must imagine yourself standing in the middle of this origin. If you want, you can even break this apart. We break them open so that each one of these becomes an independent structure. That is to say, if we break it here, this will be the 5' end. And this is the helicase. If we break it here, if this end is 3', this end here will be the 5'. So you must be able to picture it, because remember, there will be this end going this way, another end going that way. So in short, it's very important for us to just take time. You can go to the internet and understand the origin of replication and how helicase is assigned to the 5' strand. Okay. The next thing I want to talk about now is what we see at the origin of replication is what is referred to as a replication fork. Now, when the two strands are being unwound by helicase going, in opposite direction, what you have are two replication forks. These two replication forks have the DNA B, or should I say helicase, attached to the 5' strand. And on the other side, you also have DNA B attached to the 5' strand. The whole region that you see with those two replication forks, or the region of DNA that is unwound, is referred to as a replication bubble. What you have here, a replication bubble, is just a segment of DNA that has been unwound. And this segment is going to have 2 replication forks. What do you mean by replication forks? A replication fork is going to be the site on the DNA where active DNA synthesis will be taking place. So in this case, we're looking at a region of DNA that has been unwound, opened up. And we know how that happened. At this point, we want to just look at some terms. For example, when I say replication fork, what do I mean? A replication fork is going to have DNA not as double-stranded, but a region where DNA is single-stranded. And because that region is single-stranded, and we know how it gets to be single-stranded, they're stabilized by certain proteins. We'll look at them in the next slide. But at this point, a replication fork is a region on the DNA that has been unwound. Unwound means the hydrogen bonds have been broken down by helicase, isn't it? Or DNA B. Now, this region is going to have 2 replication forks because remember, it's like you're holding a thread. That if you're holding thread that is wound like in twos, when you pinch off at the middle, you're going to see that the thread is going to be opening up in both directions. So think of DNA in a similar way. You're holding a string that has got 2 ropes. The moment you pinch to the middle, you're going to find that it's going to continue opening in both directions. So now, the region where there's separation is what we're referring to as a fork. And collectively, at this region, we're going to have what is referred to as replication bubble because we've got 2 replication forks going in opposite directions. And by direction, I mean this one here is going this way, and the other one is going that way. So in this case, when we say DNA replication is bidirectional because at the replication of origin, we have got 2 forks that are present, and by 2 forks, you mean that region has got single-stranded DNA that have been unwound. Because there are 2 replication forks, active DNA synthesis can take place. And what I mean by active, I mean you can make a new strand because now the single strand of DNA can serve as a template. So DNA synthesis is bidirectional, and the principle here is that when there is a region that has been unwound, you're going to have 2 replication forks. What that means, you're going to have 2 strands of DNA that are separate, okay, and going in opposite directions. Collectively, this is a replication bubble with 2 replication forks, not 1, not 3, no. And the origin is at the center. Now, DNA is double-stranded, and what allows for that is complementary base pairing. So at this point, the helicase has managed to unwind the DNA, but it has not distorted the complementary bases that were there before. So something has to go, behind it to ensure that the 2 strands remain the way they are. And by that I mean they must be single-stranded. They must not, go back and complementary base pair, no. So now, this process recruits proteins to ensure that the strands of DNA that have been unzipped, the strands of DNA where hydrogen bonds have been broken, remain as single strands. These proteins that come in to stabilize the unwound DNA to ensure that it does not complementary base pair with other strand are referred to as single-stranded binding proteins. Single-stranded binding proteins. They do not bind to double strands, no. Similar to helicase does not bind double-stranded DNA. It means, the strands to be unwound. Single-stranded binding proteins, these are proteins that are recruited during DNA replication, and in this case, initiation, to stabilize strands that have been unzipped, to stabilize strands where hydrogen bonds have been disrupted, meaning that you want these strands to remain independent of each other. Why? Because we know they can simply go back and anneal because of complementary base pairing. When helicase unwinds, it does not distort the combination of bases. They remain intact, meaning that if nothing is stabilizing them, they'll still go back and complementary base pair. To avoid that, you recruit single-stranded binding proteins. And the next slide, I think, are just notes what these single-stranded binding proteins are. and what they do, and also because when DNA is a single strand, at this point I'm looking at point number 4, single-stranded binding proteins, in addition to just stabilizing the DNA, single strand, and I mean stabilizing, keeping it as a single strand, because when it's bound, we don't say it's, you know what I mean. So trying to say, one of the functions of single-stranded binding proteins is to stabilize DNA so that it does not complementary base pair. Another important thing that we must also keep in mind is that when DNA is single-stranded, it becomes, vulnerable. So to prevent degradation or any damage, okay, those single-stranded binding proteins come in to attach to it. So yes, one of the major functions of the SSBs is to ensure that the single strands in the replication bubble or at the replication fork do not go back and complementary base pair with a complement. They should remain single-stranded because DNA replication requires a single strand. In addition to the SSBs stabilizing DNA, another function is to ensure that the DNA, which is now a single strand, does not get damaged because now it is vulnerable. It is just a single strand. Remember, one of the reasons why RNA is very, reactive is that it's a single strand. So this DNA molecule has to be stabilized to prevent damage or sometimes even being degraded by its own nucleases in the cell. So then the take-home message here is that SSBs stabilize and protect unwound DNA strands, meaning they do not bind to double-stranded DNA. That's what that is saying. And also they stabilize and protect DNA. Now, if DNA, in this case, you can imagine a bacterial cell, it's a loop. You are the circle of 2 strands and you open up one region. What happens up front? Up front, there's going to be strain because as you continue, when you start, it's going to be easy, but as you go on unwinding, there's going to be a lot of strain, meaning that there's going to be pushback, whatever enzymes are replicating DNA, the ends up front, before the helicase gets there, there's going to be too much strain. So there must be someone who must be recruited to ensure that as these 2 strands are being unwound, up front of the helicase, the strain that the unwinding is causing is relaxed. Imagine yourself holding, strings, 2 strings that have been wound together. When you open a single patch, as you continue unwinding, they're going to create strain up front. And there's going to be pushback. Because you don't want to have pushback, because the machinery that's replicating is going to collapse, you need to ensure that you recruit someone who can come and relieve that strain. So the cell or the process of DNA replication recruits what are referred to as topoisomerases, or should I say, in this case, because we're looking at bacteria, these are referred to as DNA gyrase, whichever way you're going to pronounce it. DNA gyrase, these are a class of topoisomerases because they relieve topological stress that is caused by the unwinding of DNA. As you keep on unwinding DNA, the regions up front on both ends, remember, this is like a double-stranded molecule in a loop. There's going to be, folding, too much folding up front before the helicase gets there. And on the other side, it's going to be the same. And because of this strain, at some point, it's going to start pushing back, resisting unwinding. To ensure that helicase does not fall off, or other enzymes that replicate DNA do not fall off, do not, get the pressure from this torsional strain up front. The process recruits, and by recruiting, I mean it brings on other enzymes, or should I say protein. These are referred to as topoisomerases. In bacteria, we refer to them as DNA gyrase. So DNA gyrase, these come in to relieve the DNA of the strain that the helicase is causing. As it continues unwinding, we need to relax this DNA. To relax it, these guys come in to, to relieve that strain. And sometimes relieving that strain even requires sometimes cutting, unwinding a little bit and then, making it back, like sealing it again. In bacteria, the ones that come in to relieve this topological stress that is caused by the unwinding of the helicase are referred to as DNA gyrase. Now, let's look at the summary of what we have at the replication fork. And remember, this is just one end. We have another one on the other end because replication started at a single dot and proceeded in both directions. So what you're seeing here should also be considered on the other side of the replication bubble or replication fork. This region here is referred to, because it is unwound, this is what we're referring to as replication fork. So what do we have there? Initially, we needed someone to unzip this DNA to break the hydrogen bonds. And I remember telling you this process is energy dependent. It requires you to eat. Okay, it requires you to supply it with energy in the form of ATP. The enzyme that breaks down or melts the hydrogen bonds is helicase. Now, when these two strands are left, unwound, they can go back and complementary base pair. And when the complementary base pair, we cannot have DNA replication because replication requires that each of these two remains as an independent template. To prevent annealing or complementary base pairing, the single-stranded binding proteins are recruited to the DNA. Okay, and these proteins ensure that complementary base pair doesn't take place, one, and secondly, it also protects the DNA from destruction because now it's a single-stranded DNA, and remember it's an aqueous environment. Again, as the helicase is unwinding this DNA, upfront there's going to be strain because upfront it is not unzipped. So there's going to be a lot of coiling. And remember, this is a loop, like a circle. So when you start unwinding, it's going to be easy, but as you continue unwinding it, increasing the replication bubble, it becomes strenuous. To relieve the strain, the cells recruit topoisomerases, and in bacteria, refer to them as DNA gyrase. So this is just a snapshot of what you have at the origin of replication. Now, when we have all these things, we have got our two templates, we can now go on to elongation. So before I go to elongation, I want to emphasize everything I've been saying up to now is just DNA initiation. We know where it must start. Even if we know where it must start, someone must recognize that position. Where is that position? We said it is a 9-mer, isn't it? After recognition of the 9-mer, what happens? There's distortion in the 9-mer. And because the region up front of the 9-mer is not very weak, but it's rich in A's and T, it opens up slightly. The opening up slightly of the 13-mer allows the replication process to have single-stranded DNA. And now you can recruit the enzyme that now breaks or separates the two strands. So in this case, when you look at DNA A and helicase, we cannot say that DNA A breaks hydrogen bonds actively. No, it doesn't. DNA A causes a conformational distortion, like a strain on the DNA, and because the region up front in the 13-mer is weak, the hydrogen bonds can separate. But the enzyme that actively breaks hydrogen bonds is helicase, or DNA B in this case. Okay. And then the other things I think you have understood. Now let's move on to the next stage of replication, which is now elongation. When you think of elongation, what are we trying to say? At this point, imagine you have a single strand. And the single strand of DNA, at this point, we're calling it a template. And we know what a template is. It has got that combination of whatever pattern we need to make an identical DNA molecule. Remember, in the definition, you're trying to make an exact copy of a DNA. So this template must have the exact copy. If in a question you're given, a template, and then you're told to find the answer to what was produced from that template, it must be the same exact copy. And that is why, when you're listening to biology, some of the things that I said, you take them for granted. In the first slide, I said, DNA replication, at the end of the process, you get identical copies. But if I give you a question, I give you a template, and I give you which one of those was produced, some people will feel and start wondering, did you really understand the definition? So sometimes you need to read definitions, understand them in the context. The attitude of thinking, no, I've understood this, sometimes is what makes us miss the answer even when we can understand it. So now, I'll go back to my slide. I want us to look at elongation. Elongation is the second step in DNA replication. And elongation, what are we trying to say here? We have our single template to make the new strand or to get the pattern that we need. And here we're going to be actively adding our nucleotides. In DNA elongation, we're going to be adding nucleotides to make the new strands. The new strand is going to be made based on the information on the already existing strand, which is the parent template. And this is where now I need you to remind yourself of the structure of a nucleotide, which I won't be looking at, by the way, but my understanding is that we know. Because if you understand the structure of the nucleotide, it will help us understand that the new strand is synthesized in the 5 to 3' direction. Because when you look at the structure of nucleotide, it's got 3 parts, isn't it? And we know that on one end, we refer to it as a 3', the end that has got the OH on the pentose sugar. The end on the nucleotide with the phosphate group, that is on carbon number 5 of the pentose sugar, is referred to as a 5' end. So when we say we're going to elongate DNA, we're going to be adding these nucleotides in the 5 to 3' direction. So I hope you can picture that in your head. Well, in this section, we're also going to look at the leading strand and the lagging strand. And where is this coming from? Remember, the moment you have a replication fork, you have got 2 strands. One of them is going to be called the lagging strand, the other one a leading strand, and we'll find out why. So let's start with the enzyme. This process is enzyme dependent. It requires an enzyme for it to take place. And the enzyme that synthesizes DNA is referred to as DNA polymerase. Polymerase is like synthesizing something, polymerizing something. Okay? So, DNA polymerase, this is the enzyme for DNA synthesis. And let's not confuse the word synthesis. Synthesis just means adding nucleotides, to a template. Synthesis means adding nucleotides in the 5 to 3' direction to a template. So now, talking about the enzyme that synthesizes DNA, that actively adds the nucleotides, is DNA polymerase. But there are many polymerases. So which one of these actively takes part in adding new nucleotides to my template? This enzyme is referred to as DNA polymerase 3. Now, keep in mind, we are looking at prokaryotes, because if we went to a eukaryotic system, it will be different. But they're also referred to as DNA polymerases. In short, DNA polymerase is a common name for the enzymes that are involved in this process. And in both prokaryotes and eukaryotes, we just refer to them as DNA polymerases. But then, because this time around, we've decided to focus on prokaryotes, the enzyme that polymerizes DNA, or the enzyme that adds nucleotides to the daughter strand, using the template, of course, is referred to as DNA polymerase 3. Written like that. Now, DNA polymerase 3 is going to add nucleotides in the 5 to 3' direction. I want us to focus on this diagram. I hope you can see it as clear. For me, it looks very clear on my end, but I'm not sure about you guys, but I hope you can see it very well. So we have a template strand. This template strand is at the replication fork. That's the first thing that we must recognize here. Okay, meaning that there's another one on the other end, isn't it? So we've just got one strand. Now, the daughter strand is a strand that is being synthesized, the new strand. That's why we're referring to it as a daughter strand. The information on the daughter strand must be the exact copy of the parent strand, which in this case we're calling the template. And what I mean by that, not when I say exact, that I mean that if there's a thymine here, it must carry a thymine. No. What I mean by exact copy, it must be complementary. Because we know that when there's a T, it must pair with an adenine. That's what I mean by the exact copy. So meaning that the information that was initially here on the parent molecule should be retained, because remember, the template was not single-stranded. It was a double-stranded molecule. And whatever information it was carrying was due to the fact that where there was a T, it was best paired to an A. So if you're going to synthesize DNA, the daughter strand must carry the same exact combination of the nucleotide at that position. So if you start from the top, there's a T on the parent strand, there must be an A. The next is a G, there must be a C, meaning that the daughter strand is carrying the same exact information as the template, which is the parent strand. Now, you see that DNA, the nucleotides. So let's say, for example, you've added 1, 2, 3. At this point, you have got 3 nucleotides in your daughter strand. By daughter strand meaning the newly formed DNA. The nucleotide that is going to be added to the growing daughter strand should be in the 5 to 3 prime direction. What we see here, on this end of the daughter is a 5 prime end, meaning I've got a phosphate group. On this end, I must have a 3 prime. Let's look at the last nucleotide. I wish I could, where there's a thymine. This end of the nucleotide has got an hydroxyl group. And remember, this is a region of OH, right? So the new nucleotide, which is supposed to be added, if we're going to add the 4th one, it must be carrying a guanine. Now, the addition is going to take place, or the reaction is going to take place between the 5 prime end of this guanine nucleotide and the 3 prime end of the nucleotide already sitting on the daughter strand. When we say synthesis is in the 5 to 3, what are you trying to say? The first thing you need to appreciate is just the structure of your nucleotide. And at this point, we have already added 3. We want to add the 4th one, and the 4th one must be a nucleotide carrying guanine. What should be the orientation for this reaction to take place or for it to be added? The guanine must give us its 5 prime end, and this 5 prime end of the guanine, which is going to be a new nucleotide, must be attached to the 3 prime end of the nucleotide that is already sitting on the new strand. And because it was from the 5 to 3, we say that DNA is synthesized in the 5 to 3 prime direction. I can also say that DNA polymerase, which is the enzyme that is responsible for adding this new nucleotide, it can only polymerize, or it can only add a new nucleotide in the orientation 5 to 3. And that is why we say DNA polymerase, it synthesizes DNA in the 5 to 3 prime direction. DNA polymerase 3 is the enzyme that is responsible for synthesizing DNA. This enzyme adds DNA in the 5 to 3 prime direction. And at this point, I'm hoping that we cannot fail that question if it ever came. First of all, understanding the type of enzyme is polymerase 3. And to also understand that DNA polymerase 3 has got a 5 to 3 prime polymerase activity. Normally, we say polymerase activity. DNA polymerase 3 has got a 5 to 3 prime polymerase activity. What I'm trying to say there is that DNA can polymerize. DNA polymerase 3 can synthesize DNA in the 5 to 3 prime. It can add a new nucleotide using its 5 prime to already existing DNA. Now, one thing we should note about DNA polymerase 3 is that it's not very intelligent. It cannot initiate synthesis on its own. It needs a helper. And the helper here is an RNA primer. DNA polymerase 3, it synthesizes DNA, but it cannot initiate synthesis, meaning that these nucleotides here at the beginning, it cannot add them from the start. No, it needs a helper. And I think of it as a Dow enzyme because, it can't do that. It needs someone to help it. So what this means is that for every new strand that is synthesized, the first nucleotides are not added by DNA polymerase 3. No. RNA polymerase, and when I say RNA polymerase, it must be another enzyme, isn't it? And because there's RNA polymerase has to come in to synthesize a short stretch of nucleotides. This short stretch of nucleotides is a RNA in nature. Remember, they were synthesized by an RNA polymerase, and they're referred to as an RNA primer. Now, let me go back to those of us who do makeup. Guys, this year, like I told you, I just learned how to do makeup, so I had to buy a lot of stuff. I'm still buying some more. So I found out that before you put your foundation, this is for the gentlemen and those ladies who are like me, who don't know about primer, but it's good for you because you're going to have girlfriends and wives who need these things. So I found out that before you put your foundation and powder and everything on your face, you're supposed to add a primer. I think the ladies, if you are here, please just, give me a high five if you use those things before. I'm just learning myself. You're supposed to put a primer on the face. And what does a primer do on the face? The primer ensures that when you start putting your foundations and powders and everything else you put on your face, they're going to stick. They don't just come out. They remain intact in their place. They don't fall off. So it's like the primer, it prepares your skin to bind, for other things to bind on it very well. A primer in makeup, whatever it's called, people do makeup. They add, even me when you see me with some stuff on my face, the first thing I put was a primer. So I want us to think of a primer in makeup, whatever we do on our faces as ladies, even today men are doing it, in a similar way to the RNA primer. The powder and whatever is put on the face cannot hold well to our skin. To ensure that those things can hold on our skin very well, you apply a substance called a primer. Maybe that's why they even got that name from, I don't know. But then let's go to DNA. In DNA synthesis, the enzyme polymerase cannot initiate synthesis. And by that I mean it cannot add the first nucleotide on its own. It needs the help. So to prepare this place for other nucleotides to be added by DNA polymerase 3, a short segment of DNA, of RNA, is synthesized. I'm going to repeat. This is a short region of RNA. It is not DNA. It is RNA in nature. And it makes sense because we know RNA can complement a base pair with DNA, isn't it? So synthesizing a short primer in the form of an RNA, in the nature of RNA, which can complement a base pair, will still help us, will still help this enzyme to attach or bind, it's almost time up. Okay, let me just, I can, if you can allow me, I can just go just a little bit and then we can finish tomorrow. I hope that's, unless you're having a class. At this point, I can't see. We can continue. Thank you very much. I won't take up so much of your time. I just want to finish on this. At this point, and I'm going to emphasize something, we're saying that we have our polymerase 3 as an enzyme that synthesizes DNA. And by synthesis, we're saying adding new nucleotides. In which direction? 5 to 3. Now, this enzyme is not intelligent. It's very dumb. It cannot initiate synthesis on its own, meaning that before it can start adding these nucleotides in the 5 to 3, it needs to have a sitting, stretch of other nucleotides. Now, this short stretch of nucleotides are synthesized by RNA polymerase. RNA polymerase. And because it's synthesized by RNA polymerase, they are in nature, it's a stretch of RNA. It is not DNA. So this region is referred to as a primer, RNA primer, because these nucleotides are not deoxy, they are ribonucleotides. They were synthesized by an RNA primer. When this primer is in position, it can now offer its 3' end for DNA polymerase 3 to start synthesizing. So the take-home message here is that you should know the enzyme that synthesizes DNA. You should also be aware that this enzyme has got a 5 to 3' polymerase activity. We're going to talk about exonuclease activity much later, maybe tomorrow, but today I want us to carry home to say DNA polymerase 3 has got a 5 to 3' polymerase activity. By that you mean it can only add a new nucleotide using the 5' end of the nucleotide and connects it to the 3' end of a sitting nucleotide in the chain. In addition to that, this enzyme is not very intelligent. It needs it needs to have a pre-existing, stretch of nucleotides that can give it a free 3' OH group. In fact, we say DNA polymerase 3, to synthesize DNA, it needs a free, like free, F-R-E-E, free 3' OH group. Let me say that again because my Bemba is coming into the way I'm pronouncing things. What I'm trying to say is that DNA polymerase 3, for it to initiate synthesis, we say it needs a free, like free food, a free 3' OH group, which is what the primer does. To give it that free 3' OH group, the enzyme RNA polymerase, of course there's a specific name, it's referred to as a primase. It synthesizes a stretch of ribonucleotides. The enzyme that does this is RNA polymerase. To be specific, it's referred to as a primase. The primase is an RNA enzyme that comes in to synthesize a short stretch of DNA. It's going to be a short stretch because, remember, we need DNA, not RNA. So this guy, the primase, keeps on giving us, but we don't need that. Once the primase has done that, it falls off. Like, let's go. And now DNA polymerase 3 can start initiating synthesis. Now, let me just go to the last slide, if you can allow me. So the primase is an RNA polymerase enzyme. The primase is an RNA polymerase enzyme. What is its function? The function is to synthesize an RNA primer to start DNA replication. Okay? In this case, in fact, it should have been to start elongation, DNA elongation, because remember there's initiation that has already started. So instead of replication, it looks like it's a whole thing. No, but this time around, think of it like an RNA primer has to be synthesized first before polymerase 3 can be recruited or can start synthesizing DNA. The RNA primase is an RNA polymerase enzyme. And by this, the only thing we look at here is polymerase. It's like synthesizing, isn't polymerizing something. And it is RNA in nature, meaning that what it synthesizes is a strand of RNA. Meaning that when you're given a strand of DNA in a question, and you're asked to find, to maybe say whether there's a RNA there, a primer or not, if you inspect the nucleotides, if you see there's a uracil, then you know that the primer was not removed, isn't it? Because right now, I'm saying this and you might be memorizing. No, I want you to understand it in this context. If I gave you a double-stranded DNA and I've told you this synthesis has come to an end, but at this point, has the primer been removed or it is still there? I'm going to inspect the strand of DNA. And I know RNA and DNA, the only difference there is uracil. In RNA, I'll have uracil. In DNA, I'm going to have thymine. So meaning that if the primer hasn't been removed, it means that some process, maybe synthesis did not take place completely in full, or there's some process that is missing. We're going to look at those maybe tomorrow. But the take-home message here is that for synthesis to start, elongation in this case, you need RNA polymerase to synthesize a short primer. The enzyme that synthesizes this short primer is primase. And the primer itself is RNA in nature. So what you have here is just, kind of like a snapshot. Now, remember, we said DNA polymerase 3, it can only add a nucleotide in the 5' to 3' direction, right? So meaning that I'm going to look at, if you can see my pointer there, we are going to have a primase here. This is a primer, not a primase. The green thing here is a primer synthesized by the enzyme primase, which is an RNA polymerase. On the other one, you're going to have the same thing. So meaning that at each replication fork, or on each of the 2 strands of DNA, you're going to have a primer being synthesized first before the replication can now add those nucleotides. But of course, tomorrow we're going to see that on one of these strands, you need to synthesize more primers because synthesis is discontinuous. But today, I can end here. And here means we have understood where replication starts, the origin. We've also understood the features of the origin of replication. It has got two segments, a 9-mer and a 13-mer. Each one of them plays a different role. The 9-mer is responsible for recognition of the origin of replication, to say this is where replication must start. And the person or the protein that does that is the DNA A, which is just referred to as an initiator protein. So it's going to recognize the 9-mer based on the fact that the 9-mer has got a DNA box. And by that I mean just a binding site where they can go and attach. And once they are bound there, they cause a conformational change in that region, in the 9-mer. Because the 9-mer upstream, you've got a 13-mer which is quite weak because it is rich in A's and T's. That region kind of causes some form of local unwinding. And by that I mean a stretch of single-stranded DNA. And because now it is single, DNA helicase can come in and perform its function to unwind DNA. Why am I saying that? We should not confuse DNA A as an enzyme that unwinds DNA. No, it doesn't. Its unwinding is based on the fact that it causes a strain, the DNA, and because the 13-mer is weak, it unwinds. But the enzyme responsible for unwinding is the helicase. Once the helicase has unwound the DNA, remember those two strands can still complementary base pair. To prevent that, single-stranded binding proteins are recruited to keep those guys separate, because we understand DNA replication needs a single template. In addition to that, the helicase, as it unwinds, for some reason, it's also not very intelligent. It causes strain upfront. The unwinding is going to be too strenuous on the molecule. To relieve that strain, we have the topoisomerases, and in bacteria, we said the DNA gyrase. What do? They relieve the strain, sometimes even twisting it, sometimes even cutting it. There are different types of isomerases actually, but for now we'll just call them the DNA gyrases or the topoisomerases. Now, to synthesize your DNA, you need DNA polymerase 3. It has a 5 to 3' polymerase activity. Please go online and understand what that means in its context. You should be able to see a diagram, then maybe you can visualize it better. What it means, it can only add a nucleotide, a new one, to a free 3' end of an existing, chain or the daughter strand. This short strand of DNA, which is synthesized to help DNA polymerase 3, is referred to as a primer. For the ladies, it will be very easy for us. We can't even forget, because if you've seen people put makeup, there's a lot of mud and stuff that goes on the face. The first thing that they add is a primer to allow these other stuff you put to bind very well. In a similar way, DNA polymerase 3 is enabled to bind very well or to attach a new nucleotide because the primer allows for a free 3' OH group. Now, the primer that is synthesized is not DNA in nature. It is of RNA origin because the enzyme that makes it is RNA polymerase. And to be precise or specific, is RNA polymerase 3. So guys, I'm going to end here because I've got a class at 13. I just need to prepare for that one. So tomorrow we'll start from here, but I've sent the notes, you can go all the way so that we don't have to, Yeah, but I feel like I'm a little bit slow because I was thinking I was going to finish this. Maybe tomorrow we should try to be a bit fast. I don't know what you guys think. Yeah, because sometimes it's too slow. Thank you very much, guys. Take care and have a good day. And we hope to see you soon. I've missed the faces. I think one of these fine days we should switch on our cameras, especially for the one at 8 hours. Okay, guys. Bye for now.